Welcome to the Weekly Bioanalysis, a KCAS Bio podcast.
Hello, and welcome to the 91st episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Guarino, and I've been in the LBA and biomarker space for a little over 30 years now. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for around 30 years.
John and I are members of the ever-growing science services team here at KCAS Bio. Either or both of us are available to answer any of your questions you may have regarding this podcast or any of KCAS Bio's services. We are thrilled to have you listening to the 91st episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you're joining for the first time or if you are a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from the audience. We're constantly looking for topics, and we'd be happy to discuss something you want us to cover. So again, we're fired up to have you here, and we are looking forward to a fun episode today. To kick us off for podcast number 91, John is going to go over today's podcast topic a bit before we jump into the news and resources. John?
So, as usual, we always do some news topics, 'cause it's always nice to talk about something that we're not really familiar with. But the main topic today is the great areas of bioanalysis, approaching drug discovery, biomarkers, tissues, and emerging technologies, and just talking about some of the challenges that we have as bioanalysts working in these kind of areas as opposed to the more regulated space.
Yeah, John, great topic for today. But all right, John, let's just jump into our first news article here. Tell us a little bit about spreading to bones, cancer spreading to bones. Nasty stuff, John. What do you got?
Yeah, so scientists ID target to prevent cancer from spreading to bones. Basically, one of the issues with cancer is its ability to metastasize, going from its place of origin and colonizing other organs in the body. Bones are a common destination for metastasizing cancers, along with lymph nodes, liver, and lungs. Bone metastases can produce a near constant ache of pain and increase the risk of bone fractures. And usually with secondary cancers like this, there's not much in terms of treatment. It's just usually managing symptoms using things like bisphosphonates to strengthen bones and just working on the primary cancer rather than the secondary. But there's a team of scientists who were basically led by Lee Ma at the University of Texas MD Anderson Cancer Center in collaboration with scientists from Rice University in Houston and Shanghai Jiao Tong University in China. They basically used CRISPR to identify a protein that helps cancers to spread to bone tissue. With a long-term view of this might help figure out potential therapies to basically address this portion of cancer, so actually going for the secondary rather than the primary. And so this was just in, I think this is really just early work in cells. This one's in mice. Sorry, the next one's in cells. So basically found that mice with increased levels of acyl coenzyme A binding protein, ACBP, had a greater chance of their lung or breast cancer metastasizing to the bones. And then a further analysis of data from human lung and breast cancers found that higher ACBP was correlated with bone metastasis and poorer survival outcomes. When they actually implanted the mice with metastasis-prone human cancer cells, but treated with compounds that targeted this protein's activity, then they actually could prevent spread of the mice's cancer to their bones. And they used two compounds, imidazole ketonirastin and atomoxia. And at that point, I will take a breath to untie my tongue and pass it to you.
No, it's good. No, it's good. It's funny. I got a biochemistry's master's degree, John, and I'm proud to say I know what ACBP is, only because it's part of a lot of the pathways within the body that I had to study back in the day. even feel like I did some research on acetyl coenzyme A binding protein. It's always been a target for cancers and other things like that, specifically metastasizing. And so, those in the audience, maybe metastasis, nasty. One of the like if you get metastasis to the bone, the stomach, lungs, obviously, those are really hard to treat, John. They just,
Sure. Yep.
you can't. It's hard to get into the as soon as it enters the bone. The bones aren't really like an area that your immune system targets, right? So if you're doing these therapies, and then like the stomach, because it's just kind of meshy, like it becomes another area that's really challenging, and so not a lot of good therapies if we set it up that way. They mentioned these couple compounds here that, and John, it's tough to understand. Are they didn't treat with these compounds? They talk about CRISPR. Was it
I
in
think
conjunction with that, or these CRISPR were turning on these enzymes?
CRISPR was used to identify what way it was working. So it's here's how they did it. To identify ACBP as a bone metastasis promoter, they used a CRISPR activation screen, built a library of 2,302 guide RNA molecules targeting different genes, and attached those RNAs to protein machinery derived from proteins used in CRISPR gene editing that boosts the expression of targeted genes. The team could then insert this machinery into cancer cells and see which activated genes made the cells more metastatic. Once ACBP was fingered as the culprit, they also showed that the protein helped cancer cells generate energy through fatty acid oxidation and then avoid a programmed cell death process called ferroptosis. So then these two compounds both interfere with the fatty acid oxidation and then promote ferroptosis to halt the process. And then so that's how they did it, and so then that suggests the potential as to how to treat this in the longer term.
Fascinating. So they didn't use CRISPR to treat it; they used CRISPR to diagnose it. Right? That's new to, yeah, right. Isn't that new to us? I wasn't aware that you could use CRISPR to diagnose things, but it's pretty obvious. It makes a lot of sense that you can. But John, that's really, really cool, because most of what we focused on when it came to CRISPR, it was therapeutics, right?
Oh, sure, absolutely, yeah.
And it seems like here's the case where CRISPR was able to. I see now that CRISPR was able to get to the ACBP, right? And then through all that, they figured out these other. God bless you, John, for Temaroxin and IKE, right? Those are so they can give those. Wow, this is. Man, you brighten me up here, John. That's a wonderful, wonderful way to be using CRISPR. That's amazing to me.
Yeah. So it's like I say, it's using the gene editing and to then identify the proteins and their role. It's more not identifying the proteins; it's rather than the role, and then tagging it back to the proteins, and then saying what's the pathway, and then interrupting that pathway. So it's a really nice, elegant little process.
Yeah, and what's really interesting is that they call it ferroptosis, right? So it's iron's such an important player in the body, exactly. Right? You block that, and you can kill stuff. And tumor cells have always been known to, or cancer cells, they just sample a lot more. They need a lot more nutrients, right, John? So they take up a ton more. That's why angiogenesis is so popular. But for years, people have often targeted these other sort of iron-based things, or even sodium and potassium, are things that those cells need. Obviously, there's a lot of collateral damage if you target some of that, meaning your normal cells need it, where they must have found a way here to go through and whatever it's doing it. These things must not kill normal cells, only the cancer cell, right? So, but it's early stage, John. Great stuff.
Yeah,
great
absolutely.
research. Hats off to Rice and Texas, and even Shanghai, Tong University in China. Really clever, John. I guess. Were you? I'm going to ask you now. Were you aware of CRISPR being used as, like a activation screen? Or is this the first you had heard of this?
I'll be honest. Me and CRISPR, we get acquainted once a month while we get prepared for this, and then we generally go our separate ways. And it's CRISPR again. How you doing? No, it doesn't have a major impact on what I do on a day-to-day basis, and so I don't really see too much about it, and I'm dependent on what's out in the news to find out more.
I had no idea though, John. I've read several CRISPR articles just like you have, and you see it. I'm gonna have to pay attention now to kind of dig in to see if it's I love it. They call it an activation screen. Very carefully said there, right, John? Not
exactly.
I called it diagnostic, and they would they're nowhere ready for that just yet.
They'd
But
slap me a wrist for that.
yeah, they
Right, so
probably would.
let's
But
move on.
let's go on. Tell us a little bit more about the next article here, John.
So,
But great, that was
this is very early. This is a lab-based study carried out in cells donated by HIV patients, but it was a breakthrough in search for HIV cure leaves researchers overwhelmed. This was actually in the Guardian, and it's interesting because they emphasise in the article it is very, very early because it's just cell lines, and so if for this to go somewhere, there's a lot of work between this article and the long term. But it was just, again, it's for me, it's all about that, way people are thinking about stuff and how can we address. Yes, we're getting. I mean, you talk about HIV, and obviously we've got the medications like PrEP, etc., where it's preventative. It drives people's counts down to really low levels, so they're living with HIV and it's not in real danger of it going much further than that. But it's not a cure. It's just purely keeping it under control. But this is a team this was in Australia. And it was the Peter Doherty Institute for Infection Immunity in Melbourne found a way to make the virus visible, to paving the way to fully clear it from the body. Because part of the problem with HIV is it conceals itself inside white blood cells, and so it means that there's that ability means you're not fully eradicating it, and there is this like long-term reservoir that could, obviously, if you stop taking the drugs, could then reinitiate and progress further. So this is an mRNA approach, where what they did was they developed a new type of LNP, or lipid nanoparticle, to actually deliver the mRNA to then, if I can find it, I can't find what they did. They basically, it's sorry, let me.
Yeah,
They basically they delivered it to really, here we go. They shown for the first time the mRNA can delivered into the cells where HIV is hiding by encasing it in an LNP, and then the mRNA actually instructs the cells to reveal the virus. That's the mechanism behind it, and they did it once and they weren't sure it had worked right, so then they've done it multiple times, and the results always come up the same way. And like I said, the key to this is more the delivery of the mRNA to actually get it into the cell to then expose the HIV, to then hopefully then open up treatment mechanisms. But it's all very early stuff. But it's interesting. It's not always about the treatment. It's like, in this case, it's the delivery is the key to then help actually possibly give you the treatment pathway going forward.
Yeah, they mention here it's a fascinating mechanism, John. I'm still trying to tease it out here, but it looks like the HIV itself is sequestered in some. When the HIV is sequestered, this little LNP forms a fat bubble around that somehow, right?
It's around the mRNA,
right?
It's basically, it's around the. I mean, I assume it's typical sort of LNP mRNA delivery mechanism that we're using in all sorts of different therapies. But this is, like I say, a different approach to get it into this specific environment, different LNP.
Yeah, because LNPs typically don't get taken up by white blood cells, but they found a way to overcome that, right? So they can deliver this payload. Deliver the payload to white blood cells, and then, which has always been pretty challenging for a variety of reasons. White blood cells typically don't take up the LNPs. They're also kind of turn over pretty, not like red blood cells, but they have some turnover to them. And then the fact that it then exposes it, right, John? So somehow it gets in there, delivers its payload, and now these cells start to clear the HIV, which this is historic, John, right? So you kind of touched on it early on. There's been cocktails, and now I think you can do that cocktail treatment in a single pill, right? I think they've really kind of in the last 20 years refined that. It's not, but it's still, 40 million people are taking meds to suppress, circulating HIV levels and to keep your CD4 cells high. That's what all that cocktail does.
Yep, sure.
And so if you didn't know, HIV, in order to go from HIV to AIDS, right, there's a difference. You can be HIV infected but not have AIDS, right? AIDS is
Yep, sure.
less than 200 CD4 cells, which means the virus is sorry, I said CD4, there's CD8 cells, but the virus or it is CD4 cells, excuse me. The virus is knocking out all your T cells, your helper cells, and then your immune system's down. But this, John, it is, I mean, wow. That's what I got to say to this, John. Like, wow, this really, because there's some other authors, they kind of quoted other institutions that are like, yeah, no one's, there's been a lot of therapeutics that have gained some traction, but they never make it into the clinic for, maybe they have the delivery of it might be difficult. Like for a little while, people were trying to go with vaccinating or treating like intraanal, right? That's not going to work, right? You can't do 40 million of those. So you got to find a way to deliver the drug that's reasonable, or even vaccinating that way. But this seems like a major breakthrough, and looks very, very promising, especially when someone says this isn't right, go back and do it, and now they're like many, many, many times, and then it looks like another university has recapitulated it, right? Is that it looks like Oxford or somebody?
I think it's certainly this
Francis Crick Institute. It looks like right there.
I think he's just saying that the guy at Crick is basically saying it's like, do you need to eliminate the entire reservoir for success or just the major part? If just 10% of the latent reservoir survives even this approach, will that be sufficient to see new infection? They don't know. But again, it's a new approach. It might just give that path forward. And actually, Michael Roche, who was part of this University of Melbourne team, who's one of the senior authors of it, said it could have actually broader implications beyond HIV, with the relevant white blood cells also involved in other diseases including cancers. So, again, it's one of those things. It's early research. It's a long way to get it to humans and beyond. But
it looks like there's a Debbie Down. Yeah, go ahead.
Like, as we've talked so many times, research depends on someone making this finding, and this group will build on it, or other people will build on it. Eventually, it will lead to something. But so it's I mean, there's definitely a lot of potential here.
Yeah, there is one professor here, Thomas Hunke from. I don't know if I said that right. He's a little bit of a downer, isn't he?
Yes, absolutely.
Yeah, I was going to call him a Debbie Downer, but I think it's important to just maybe say it real fast, and we can move on to the next one. But like, it's not. We don't know. We don't really know if HIV is infected in every cell and every tissue in the body, John. Right? That's what this individual's saying, right? And there's a good chance. Obviously, if your white blood cells aren't being depleted, then you can. There's the latency. HIV being latent in your epithelial or endothelial cells, who cares, right? Because it's just in there. It's the latency and activation in your white blood cells that causes this AIDS to go down. So I don't know, but he said it's still a pipe dream to think that, or it's merely a dream that you can affect every cell in the body. John, I used to believe that maybe 10, 15, 20 years ago, but these CRISPRs and other technologies, I think we're going to get to a point where another 100 years from now, there's a good chance they will be able to target like every cell in the body with some sort of, kind of lock and key system, right? Where only cells that have certain things will be able to take up any sort of message. And if it were an mRNA thing, where others will not, I think we're getting there, John. I really do. Where, in the original cancer therapeutics, right? And we still use some of those affected every cell, right? And that's why people lose their hair and things like that. But I even think those medicines with the advancements of ADCs have come a long way. But now I'm taking us way out there, John. But I got Star Trek in my mind, where you walk into the doctor, they use a little beam on you and you're all set, right? Like, that's coming. I feel like it is.
But you think how quickly all this has moved in just our lifetime, and it's not even our lifetime. It's a part of our lifetime. I mean, the speed at which some of this is moving. I think all of it's possible.
John, I mean, it's
to build on,
Yeah, I mean, you're right. In our lifetime, I'm going to quickly tell a little story here. In '94, when I first did my first autologous T cell therapy, it took 14 days. They got those things down to 5 days now, and now the science has come so far. It's not an autologous T cell or allogenic T cell. It's not. They don't even need cells anymore. Instead, now we're using LNPs or AAVs or some sort of mRNA type of deal, where you use the body generates it, right? So that you vaccinate with that, and the body then makes the CAR T cell. Man, that would have, if you had told me that 30 years ago, I would have laughed at you. You know what I mean? I would have said, "Oh my God, you're pipe dreaming." So to your point, that has come pretty dang fast. I hope to see what happens in another 30 years, John. But let's go on to the next one. Great stuff, John.
Right. This one is small molecules are still relevant, New compound kill parasite that causes Chagas mice disease in mice. Scientists have identified a new class of compounds capable of partially clearing the tropical parasite that causes Chagas disease out of mice, suggesting the molecular group could serve as a source of new treatments for one of the deadliest parasitic infections in the world. What they actually they come up with a number of compounds called quinazolines. Their lead compound reduced the number of parasites in the blood of infected mice by 81%. Now, here's the catch right now. Although they're active against the parasite, they don't last long enough in the body to make effective drugs for humans. Well, I read this and I immediately thought the parallel to GLP-1s, where it's all based on Gila monster venom and the actual native Gila monster saliva. Sorry, it's all based on the native proteins that they know what they want to do, but those had a problem with therapeutic window, and so they weren't really useful as they were until the group at Novo Nordisk designed the GLP-1 with the long lipid chain to increase its half-life in plasma. And now look at where we are with GLP-1s. So it's a first step. So they basically right now, that compound that they identified as their lead is actually this work is mainly been done at the University of Dundee in Scotland, and now there's a separate group at De Montfort University in Leicester, which are now looking to make more stable forms of this to hopefully then give this a forward path to really help address what is a nasty disease. It's caused by protozoan Trypanosoma cruzi and infects about 7 million people around the globe, mostly in Latin America, killing more than 10,000 every year. So infection, you got fever, headache, difficulty breathing, and chest pain. And this was interesting to me. There are 2 approved treatments for the disease, but they cause really bad side effects. And 30% of survivors develop a currently untreatable chronic form of the malady that causes heart disease and an enlarged digestive system. So that's not cured. It's obviously like a long-term major problems, and the toxicity of the approved drugs is so severe that in most cases people can't complete the treatment regimens. So I'll pass it to you, and then we'll obviously have more.
Too much to add, John. This is a little bit. You're talking about this tropical parasite, John. I'm not. I'm trying to. Man, I took a parasitology class a long time ago. It feels like. And I can't even remember what is this, like Chagas disease. 7 million seems like, but like, what does it cost? Is it like inflate your liver? Okay, symptoms of fever. There it is, right? Like I didn't look. I don't know much about this, but again, it just gets back to small molecule is not going anywhere, right, John? That we
exactly,
can't that at all.
Yeah.
And then when it comes to pretty nasty, like the current anything that when you read like 2 approved treatments, as you mentioned, and they cause like heart disease and enlarged digestive system. Yeah, I don't want to take that. You know what I mean? I don't know what Chagas disease does. Maybe it kills you, but that sounds really bad. I don't want those things. So, and then John, I can't. These are it's targeting some enzyme, right? Is it the lysyl tRNA synthetase is what they're blocking? Is that right?
Yes, that's exactly it. It's sorry, they did some fairly, again, this systematic approach to try and identify the pathway. So basically they used they grew many different populations of a related parasite to the Trypanosoma cruzi, each with a different gene tweaked to produce more of its protein than usual. Then they added the quinazolines into each of these parasites to see which ones could cope with the quinazolines and which couldn't. And so the ones that couldn't, they were figuring out what was the gene responsible and which coded for an enzyme called lysyl tRNA synthetase. And this enzyme is critical for the parasite to make proteins. It's loading the lysine onto tRNA, so then it can go into developing protein during protein synthesis. Without lysyl tRNA synthetase, the parasites can't incorporate lysine, and so then the protein synthesis essentially grinds to a halt. So that was how they figured it out. That was how they screened and then figured out the mechanism. And it's almost like it reads that they had the drugs as potential drugs to. Oh, they must have. I think they must have like halted it with the drugs, then step back to figure out the mechanism. And so that was how they had the quinazolines as that agent that was going to basically halt the parasite. This group are just really focused on this kind of approach for tropical diseases. But an underlying issue here is what we see. But this is the line that actually jumped out at me. We talk about this nasty disease. We talk about drugs that have got horrible side effects. The disease kills 10,000 every year, but it could also increase in prevalence as global warming is predicted to allow T. cruzi, primary vector, that are insects, commonly called kissing bugs, to expand northwards. And that's something we see in the States with, mosquitoes with West Nile virus coming further north. We've seen, obviously bees coming up from South America. I know before this we were talking I've seen migratory birds. They're changing their patterns. I went back to when I was back in Scotland a couple of years ago, and when I'm in Glasgow, I love to sort of swing by the house I grew up in. I lived there for 18 years, so it's just nice to see the village and how the house looks and everything. Yeah. And I was like, but we had this big laurel hedge in front of the house, and there's all these long-tailed tits running, basically flying in amongst all in the hedge and the cherry trees behind it. And we never saw those when I was a kid there. They just never existed. They were much more of a Southern England type of bird. They'd be there in the summer, but to see them in Scotland was incredibly unusual. Well, it was, but it's now changing pattern. And as we were talking before this migration of animals could happen historically. I mean, so... I'm going off on a little bit of a tangent here, but it's really interesting because it plays into all of this. A few weeks ago, there was a restaurant in the US did a thing called Green Crab Week. And the green crab, as I knew it growing up in the UK, I knew it as the shore crab. And actually, I knew it as more than that. But the father was a marine biologist who had this horrible habit of referring to everything as its Latin name. We never knew it as a shore crab. We knew it as a Carcinus manus. My sisters and I still refer to it like that. But anyway, this crab came to the US in like the ballast of sailing ships. It's been here for a few hundred years, but as the waters in the northeast have got that little bit warmer, the shore crab population has exploded, and so it takes out native fauna. It's a voracious hunter, but obviously in Europe and the UK it's controlled, but here it's not. It takes out things like eelgrass and stuff that are soft-shell clams that are vital for other organisms. So people in the northeast are thinking, what can we do with these crabs? So this was the basis for Green Crab Week and restaurants coming up with dishes to sort of try and use this crab. So the restaurant in our village called Hazelnut Kitchen, which we go to occasionally, they actually were participating in this week, and they did this really lovely pasta dish using green crab as one of their main ingredients.
Oh, there you go, John. When there's an invasive species, we eat it.
It's a lovely tasting crab.
Well, maybe they start eating the turkey vultures over here in Kansas.
So it is like how at least it's one prong to address this issue, but it's an issue. I mean, like I say, as insects migrate northward, work like this at University of Dundee is actually really, really key, because there will be these illnesses that we never had to worry about. But as you know, as the pattern of organisms change in terms of migration, or they just the range increases, then we have to think more about it. I mean, I'm in the Northeast, so I'm pretty safe at the moment. But I love seeing work like this because it's telling you people are thinking, okay, we're okay now, but what are we going to be like in 30, 40, 50,
Wow,
60
that's good.
years' time, you know? And
Yeah.
it's really, really interesting work. Anyway, I will get off my
Yeah.
soapbox, but I love this stuff.
No,
It's
it's
just
good to listen to you. I mean, you got me thinking about like how I do. You know, I go by I grew up in a home for a similar time, John, about 22 years in a house, and I drive by it periodically. I'm going to start looking if the vet. It definitely looks smaller. I can tell you that it looks
sure,
a lot smaller.
yeah, yeah.
But it's interesting to see if the vegetation's changed at all. And last time it had, they cleared out a lot of the woods around my house. And John, you know, the thing that we couldn't be further removed from this. This is the most basic research, right? You said it.
Sure,
These are
yeah,
guys in Scotland looking at diseases in Africa or South America, right? Wonderful. I mean,
yeah.
we're on the spear of the drug development end, trying to get these things over the finish line, right? We do drug development here, right? That's essentially what, to me, a lot of bioanalysis is, right? And these individuals are over here searching, and what is great is I think it has good. It's not just they talk about other like fungi, and other
Yep,
things that they
yep.
can use this mechanism on, just to bring us back. So
So
it's a great story though. And small molecules are still highly used, and you know they're curing diseases like never before. And I got to admit, John, it's definitely, like you said, it's the reason why I think I love the proactiveness of it. You kind of
yeah,
said it. I'm in the
yeah,
Northeast. I'm safe, but none of us are really safe. That's why we do these things, right?
yeah.
So, and remember, we had a big scare. I felt like we had a scare with, the antibiotic resistance and MERS and all that, which we seem to have broken through a little bit. You don't really see as much as that now. It wasn't. Seems like there's been some breakthrough drugs to help with that. But anyhow, maybe a good news article.
But I would always say, even with something like that, there's cycles to it. So it's like
Yeah.
it's always us against the infecting organisms. So we might have, say for example, we have antibiotics that we have the upper hand to, but it eventually it could mutate, and then we're trying to think, well, what do we do now?
Yeah. No, but I think that's why we're staying ahead of it too, right? I do think
Yeah,
there is that. It's definitely stuff we want to research. I mean, basic research is needed. John, do we want to cover this last article?
The last one
Yeah.
could be really, really brief.
Yeah, let's
because
do it. We got a light agenda. Let's go ahead.
Yeah.
Let's go ahead and talk about this one.
I thought it was. We don't even need to spend long on this because it's actually just a follow up to a previous podcast where we talked about individual CRISPR cures, where they had the child called KJ Muldoon, who became the first patient to receive a personalized CRISPR gene therapy, to basically address the genetic mutations causing his rare metabolic disease. And so we talked about this for a long time, and I'm not sure, I should have checked which podcast it was, but it's fascinating because it was basically a team came up with an approach, but lots of people also chimed in to help drive this particular CRISPR for this child. Eventually he was able to his disease was urea cycle disorder called carbamoyl phosphate synthetase 1, whereas he lacked the enzyme to process ammonia during protein digestion. And so basically they came up with a CRISPR approach to help that, and so he will likely have the disease but much milder form, but he has been able to go home and live with his family. But the key for that was you had the group who basically came up with the approach, but lots of other research groups chimed in to help with it. And actually what has come out of that is there's a new $20 million effort from the Chan Zuckerberg Initiative and Innovative Genomics Institute to ensure that this isn't a one-off. So they can actually, they've launched a new center to create custom CRISPR for kids. So they called it the Center for Pediatric CRISPR Cures. Will bring together the gene editing expertise of the University of California, Berkeley, with the clinical know-how of its neighbor, the University of California, San Francisco, to develop cures for infants with severe genetic diseases. your 20 million funding for the centre comes from grant from CZI, the Chan Zuckerberg Initiative. So I just thought it was a really feel good story in terms of the cure, the science, the collaboration, and here's the next step, and it is, moving forward.
Yeah, John, it's a wonderful story. It's a great philanthropic story. It sounds like, and I didn't realize, this is surprising to me, that the Innovative Genomics Institute, IGI, right?
Yep.
And that's Donna Hara, who's that's a different individual, but that institution's been around since 2015, and it is the Nobel laureate Jennifer Doudna, who is
Doudna, I
the
think it is.
founder, who's the founder of that. And it's John, I didn't realize how strong UC Berkeley, UC San Francisco, and UC Davis were, because that's a 10-year initiative, right? If that was founded 10 years ago,
Yeah, sure.
that just, I don't know why that's so surprising to me. Sometimes our CEO John will say it took 10 years to be an overnight success, but I mean, obviously CRISPRs have really hit the stage. They're in the clinic. They're still in the early stages of the clinic, but I'm sure, to me, it seems surprising. I don't know why I'm surprised by 10 years of that institution being around, but it's a great story, John. And certainly, I mean, when you see people born in 2024 being treated with things, it's like, man, that is really, that's amazing to me, that they were given this treatment to help with some sort of rare disease that causes a lack of your ability to process ammonia, which that kind of happens a lot. Well, not a lot, but there's other diseases that affects. So wonderful stuff, John, and that kind of wraps up our news and notes. Unless if you had some parting remarks, but really, really cool stuff coming out of California with CRISPR.
I think, like I say, all cool stuff. Hats off to all the people today in terms of what we've talked about. Yeah, so let's move on to the main topic.
Yeah.
And so we thought, as we've had a number of guests on recently, it was about time we just came back to the two of us.
Sorry to our audience for that. No.
Gibbering about a subject we chose at random,
Yeah. Well, it's a good one.
It's something that we spend a lot of time talking about, and this we just as a title, it's the great areas of bioanalysis, approaching drug discovery, biomarkers, tissues, and emerging technologies. And these are things that can be, when it comes to a validation. So the easiest thing for us is validation of a PK assay. Someone comes to us with a drug and said, we want you to measure the amount of this in plasma. And so you come up with you develop a method, you then validate the method to an SOP, so all the experiments are going to go into that validation, and it's a very prescribed process. It's easy. It's easy. I mean, we know what we're going to do. You know the goal, the means to reach the goal, and you know what has to be done in every step of that. For each of these categories that we listed here, drug discovery, biomarkers, tissues, and emerging technologies, you're taking away some of the surety of that. It's not as definite as what we want to develop. We want to develop and validate a PK assay, and it ends up with a lot of discussion. I mean, we had a training session last week that Lindsay Rutherford did, which was great in terms of protocol interpretation. But we touched in that on just fit for purpose validation versus qualification. And when Lindsay had said at the beginning of the training session, I'm not sure this will take the full hour, we overran because this topic
famous is last words, right?
For us. And so it's like so it was actually, the timing was perfect, because it got me thinking about, like I say, we came up with the topic at random that we knew we wanted to talk about, but it did sort of help calibrate some of that in terms of this is how much it affects us on the day-to-day basis, or affects bioanalytical people, and so that was that because as we step into any of these pieces, drug discovery, biomarkers, or pharmacodynamics, tissues, and like these emerging technologies, it needs a discussion with the client about what we actually need to do to meet their needs. And sometimes we way overscope it. Other times we're underscoping it.
Yeah.
But we're dealing with that, trying to find our feet to basically help deal with it. And I just thought it might be quite good. Might be interesting to walk through it. And we'll give our perspective on some of this, and so that was where we came from.
Yeah, no, John, nice job teeing that up, right? I like the way you call it the gray areas of bioanalysis. That's very true because, as you kind of alluded to, like there's pretty straight. If someone comes and says, "Hey, I need a PK assay," or even an immunogenicity ADA
Sure.
assay,
Yeah.
there's two documents out there you can go get that give you great guidelines on how to do that, the validation of it. Even white papers out there if you have some challenging drugs or maybe even a challenging matrix. But when you're going to do PK and validations, PK and immunogenicity, it's pretty clear cut, John. If you don't have if it's not working, you got to go create a critical reagent to it. But over time, you're going to meet those criteria, right? It's the accuracy and precision within 20% or 15% in your world, right? Maybe 20% in LBA world, right? So those types of things, and then selectivity, right? Specificity, all those things are pretty straightforward. But then, as you
I'd also like to say something, because you mentioned it. We have the documents we can refer to, and this is because of years of industry discussion coming up with those documents. Because in the early days when I was doing LCMS, we had our SOPs at what were the company at that time were called Advanced Bioanalytical Services. We had our SOPs, but we were particularly because we're doing a lot with large pharma, they all had their SOPs, and often it would be a negotiation about which set of SOPs we would use because our acceptance criteria were actually different. And so it's like some would place more emphasis on calibration standards, others QCs. Just, stability, you'll be within 15%, or do you measure it versus time zero versus nominal, all this kind of stuff. And so there was a lot of toing and froing, but obviously there was a lot of industry discussion to standardize that, which has got us to the point where validation is, yes, this is the way we do validation. Here's what we do. Here's the criteria that are universally accepted, although the moiety that you're working on may require wider criteria because of the complexity of the assay, etc. But it's still very prescribed, and even if you widen the criteria, it's not a big change. You're still going to basically do the same kind of process. So I'll pass it back to you.
No, John, and you if you as an LBA expert, we were trying to put a square peg into a round hole because we were taking what the small molecule kind of PK kind of evolved into, and I kind of lived through that. I've been working here so long that when I first got here, there was still a lot of what I'll say commingling, where people were like trying to take a PK small molecule LCMS approach to a ligand binding assay. Now that's all been sorted out, which is lovely.
Sure,
that was a mess for a little while. I think there was just the LBA assay just had somewhat different criteria to give you comfort that it was robust, right? Where a mass spec, some other things carry over, right? Things like that. They don't exist in LBAs, but it's interesting that that's happened like in our own lifetime. And then you fast forward to today and the topic we're talking about, it's like you mentioned drug discovery, right? If we get into that, there's a lot of people when they start to do early drug discovery, they're putting maybe 3 or 4 candidates into some sort of rodent or some sort of animal. That assay there, right? That oftentimes people want to, I don't know, qualify it or even look to make it pretty robust, and that's the gray area we were talking about, John, right? And that's where, as a CRO, we find ourselves, kind of going in like, well, what are you comfortable with, right, John? You've heard me talk about, well, we're on the highway together. Are you going to go 65 because that's the speed limit over here, and then, or are you going to go 55, right? And that's and what that translates into is if you're going 65, then you can do a discovery method that's pretty rough and ready, right, John? And we can talk about what rough and ready is in a minute. But if you want to go 55, then maybe you qualify it, right? Yeah. And then some people, unfortunately, they want it to be like qualified at a validated level, and that's where they get. To me, that's where they get into, I don't know if trouble's the right word, but it's just too much, right? You're over baking that is what happens. You just don't. It's not. You got 3 or 4 candidates. You're just trying to get some rough PK profiles, and you don't have to make heavy investments in specific critical reagents so that you can get the sensitivity and the specificity of a validated assay, right? So
And
that's, but I'll take a, we're going to talk a little bit about how much, the advancement in what I'll say early discovery, PK, and even immunogenicity assays off the charts, John. At least in my world, those reagents, those capture reagents, weren't readily available. Now we can go out and buy 5, 6 different, and I'm kind of being specific to maybe like ADCs and monoclonal antibodies, but there's plenty of proteins where you can just find a commercially available, often you can find a commercially available antibody against, or a capture reagent of some sort, whether it's an antibody or the target antigen or something. But that's come a long way in the last 10 years. And so that allows you to actually be able to do an early discovery assay that's kind of in between, right? Maybe it's not like going to meet a pre-validation or validation criteria, but it's still better than just rough and ready. And I'll pass it over to you, John. But that's a fascinating change in the last, say, 5 to 7 years, in my opinion. Almost.
And I think for me, the internal sort of tension with early discovery, I think as bioanalytical LCMS people, our tendency is always to want to give that assay. We don't necessarily want to spend forever developing it, but give something that is really, it's far better than it needs to be. We talk about throwing QCs in to qualify it. We want to do benchtop freeze thaw again. It's like we're starting to add these things. We really want to show our assay is really really good.
It's always mass spec.
It doesn't need to be really really good.
It's funny. It's you mass spec people that want more. I was just thinking about that. It's the only time we've had issues with, and I should say it's not a lot, but when people are like, "Hey, I want more out of my discovery," it's usually in the small molecule, because we're like, I think the LBA people just more accepting of it. But go on, Joe. Sorry to interrupt you there. Yeah.
But I think when we talk to people in the really early discovery piece and we're scoping, I think we have that tendency to say, well, you want to show the assays working, you want to show that it's doing what it needs to do, and like I say, we need to be listening more to the client to hear. I just want an assay to say what's the trends, and so obviously you want to show that you're extracting right and your chromatography and your mass specs all good, but ultimately I think we still have that tendency to go further. So we're trying to. I think there's always an education to try for these very early opportunities. Put a break on yourself. It's the bioanalytical mindset, Anyway, that's what I see as the tension there, and I think we're getting better at it. But we certainly. I
still think there's some legacy LCMS people that aren't nearly as comfortable. I mean, in the world of ligand binding assays, we're getting real comfortable with rough and ready discovery PK assays, where we do like a day or two of development. We get the range of quantitation. We understand the minimum required dilution. Maybe we take a peek at dilution linearity, and we're off and running. Yeah. And sometimes the background might be high, but it doesn't matter. We got a suite of reagents that usually work. And I'm kind of talking about more like generic, generic monoclonal antibody assays, or assays for mAbs and ADCs and ARCs and things like that. But also, the ADA has come a long way too. Like, you can do a rough and ready ADA. Like, again, it's just put the antigen of the antibody target down and really just do signal to noise now, and no positive control. I mean, these are things that it's that lean approach as well, for even swerving into the regulated space where people are looking at, or even for support of a tox study, maybe not validating all 3 tiers of an ADA assay, but doing a qualification for 2 tiers is sufficient, and it can still be part of a regulatory filing, because it's answering your questions, right? That's really what's it about. Can you answer your questions?
Sure, that's exactly
the hypothesis, right? We're going to talk science speak. Is the hypothesis proven by your data? But yeah, I feel like the field is becoming more comfortable. And by the way, John, what's it doing? It's accelerating science, right? And
sure.
That's what we do here at KCAS Bio. We're science accelerated.
And it's like that, that like you said, the generic mAb approach, you do it with LBA, but also we do it in LCMS now, because again, it's that that getting the mindset right, and it's also having the customer who wants to take that, who wants someone to help with taking that approach. So, as a CRO, you don't necessarily go in that direction until you got someone to
John,
collaborate with, and it
always
really helps drive things.
Yeah, it always seems like it's someone in mass spec who's like, "Oh, where's my internal standard or analog and things like that." When you're like, "Wait a minute, you wanted a discovery method, right? Like you bought that." No,
It's good, but I mean, that is a debate you
because
will
they say,
have.
"Oh, I needed to submit this." You're like, "Well, it's a
Yeah,
non-GLP tox. You can still submit it, right? You don't need a validated method for
but
a pre-tox or like I said, a non-regulated GLP tox study."
so,
But
so Jeff, I just step in. The key with LCMS is, yes, it's a great sort of technology in terms of bioanalytical, but because you're doing adjacent wells that are all going through the same process in parallel, it is really useful to have an internal standard to compensate for that variability between wells. It will tighten things up a bit, but
usually we can
again,
find one, right? Well, I don't want to
again,
make it sound like we're like, we're just winging it over here at
no, no,
KCAT Bio. No,
no,
most of our discovery methods have some sort of
sure.
analog to them, right?
So yeah, what I'm going to say, yeah. So again, because when we're in validated or validation, well, we really want stable label internal
Oh,
standard.
absolutely. Yeah.
But prior to that, you can be thinking all kinds of flexibility. We have a list of generic internal standards that you could all throw into, say, just one analyte and see which ones come close and which tend to behave similar to that analyte. Or if you're a little bit further into discovery, then you're into the realm of maybe having an analog where it's structurally similar, so it's going to behave somewhat the same. And honestly, if you can, I think when it comes to taking an internal standard off, that's where you probably have the most pushback from. It actually comes from customers,
Yeah.
even because I've had that with assays in the past, and where it's like it's a pretty generic approach, but the internal standard isn't behaving like the analyte, and you say, but if we take the internal standard off, the curves look great, standards look great, QCs look great, this could work. And I've had numerous cases with clients says, I'm not comfortable with that. I need you to figure out how to make it work with an internal standard. And it's like, it's working, So
Yeah.
there's obviously a to and fro. That's where the conversation between the CRO and the client is really important on all stages. But it's true throughout the process. But that's one of the ones that we will definitely have.
All right, John. So yeah, I want to make sure we cover all the things in our agenda
absolutely.
here. So I feel like we close the loop on the gray area and drug discovery, right?
Yep.
But let's maybe move on to some biomarker tissue. I want to go biomarkers, right? That's a hot topic these days.
Do whatever you. So we. I mean, we can talk about qualification, fit for purpose validation, biomarkers.
Yeah.
You talk whatever you want. Yeah,
John, I think that's one of the biggest controversies in the bioanalysis world, right? I want to be real dramatic, John, but there was a lot of publications on AAPS threads in the last month or so, maybe 3 months, and a lot of discussion about a qualified biomarker versus a validated biomarker. And I think the real confusion is you got to set the tone the right way.
Mm-hmm.
A lot of people are talking about it clinically. And what I mean by that is something a physician might look for, or a reference laboratory might offer a test for. Those types of validated biomarkers are they're talking about it clinically, and
Sure.
then if they're doing qualification, that means, hey, we don't know if this biomarker is relevant for said disease, so we're going to qualify it and see if we can confirm it is a validated biomarker. That's all clinically. That doesn't mean anything to you and me over here in our bioanalytical world. We got completely different words for that. So when they talk about somebody's doing biomarker qualification clinically, they're screening thousands of proteins and compounds and things, and they're looking for biomarkers. And then if they say, hey, we're validating this biomarker, that means they're proving the hypothesis that these biomarkers are relevant for this neurological disorder. Think about that, like TAUs and all those, or things like that, where people were for years looking to clinically confirm that these are validated biomarkers for neurodegenerative diseases. That speak is not what we mean when we talk about a fit for purpose validation versus a qualified method, because everything in the biomarker world is context of use and done. It's
Yes,
done fit for
yes,
purpose validated, and it gets confusing because presumably the biomarker of interest has already clinically been validated, John.
yeah,
See that?
yeah,
That's an
yeah,
important piece. And
yep,
so then bioanalytically, you can say, hey, I did a true fit for purpose validation and went on to try and we validated that assay, and we can talk about what that means. Or really, I did a fit for purpose validation, and all I did was run a kit once and confirm that the kit performs as the range of quantitation is good, the MRD is all set, and the dilution linearity look good, and now that thing is for its purpose validated, and all I
yes,
did was
exactly.
develop the assay,
Yeah,
right? So
yeah,
that's the piece that is, I don't know if I'm gritting my teeth a little bit if you can't hear it, because that gets confusing. People want to eliminate the word qualified from our world, and I feel like that's a danger, because when I think of a biomarker that's been fit for purpose validated, or you talk about having it bioanalytically validated, that means it's gone through QA, John. And,
yeah,
and we
yeah,
like to use the word qualified to really let it know, hey, I can send you a biomarker qualification that's a 300-page report, but it just didn't go through QA. It's an incredibly robust and sophisticated thing, but it just didn't require to have to meet the rigors of a validation, 20%,
yes,
errors,
yep,
some sort of other sort of, I didn't do necessarily 6 ANPs, although you can validate a biomarker and still do 3 ANPs. So that's a gray area right there, because I might think you need 6 ANP runs because, but really you can validate, you can do a true fit for purpose validation or a GLP validation. Actually, it's a GCP, right? We don't really validate preclinical stuff. So it'd be a GCLP validation, and it could still just be 3 ANP runs, and that's going to be acceptable. I
yeah.
don't I don't
Whereas
like it, John. I want to do 6, but that doesn't mean you can't do it that way, right? So I'll pass it back to you. But
Whereas, whereas we
this is
will always
becoming
do
a
we'll
yeah
always do 3, but then people will say, "I don't want you to do 1," and then it's like
yeah.
because yeah, it's like oh, then you got that debate in that direction. I think
And I'll say
that's
this:
where
we're
I
not
see a
concerned.
lot of confusion
Yeah,
between us and clients is when
and
we're talking. We
we're
want we
John.
want
Go
we
ahead.
want to qualify, and they say, but we want to validate, and then you say, but what? Then it's like, well, what is your validation? And they sort of say it's just maybe one accuracy and precision or whatever. It's like, but that's what we would call a qualification. So you have this disconnect in language, and sometimes that's people who aren't necessarily bioanalytical, but they have a project that I particularly like to say with a biomarker, and so where we're saying bioanalytically qualified that we know what that means in our head. That doesn't they don't can't compute that language, and so then they say, "Oh, we want it validated." So you have to say, hold on, what do you actually mean by validation for this? Because here's what we mean by qualification. We can call it qualification, fit for purpose validation, whatever you want us to call it, but here's what we are considering doing to get this, to show that this assay for this biomarker is sufficient for your needs. And that's the key. It does come back to context of use. Like
Mhm.
it's a huge part of the conversation. I remember EBF were really big on this several years ago, talking about you don't want to fully validate biomarkers. It doesn't make sense. Talk about context of use, and then what kind of assay do you want to develop, and what experiments are necessary to show that that assay is suitable to meet the needs of your biomarker. And that's the key conversation. But that becomes that's works really well between bioanalytical people because we're thinking the same. But once you're outside of that, then it becomes a little bit more confused.
Yeah, no, and John, the term, when you think about there's a big push in the field now, and at AAPS Summer Scientific Forum, I was approached, and they're trying to sort of start a group to kind of normalize the EU and the US with biomarker vernacular, okay, nomenclature,
Mm-hmm.
however you want to
Yep,
talk
yep.
about it, speak. And to me, it's like the solution that I'm proposing would be to meet both places, because the EU, they staunchly believe you shouldn't be talking about the word qualification shouldn't exist in biomarker speak. And again, that's tough for us over here because in the US, because it really, to me, it's like, hey, the word qualified really sets my mind in terms of what you did. If you just say it's a fit
Yeah,
for
yeah,
purpose validation and you show me one A and P batch and it didn't go through QA, I'm going to look at you and think, do you know what you're doing? Right? That because, but what solution I think would be good is everything's. I was talking about it, alluded to it earlier. Everything's a fit for purpose validation, but you have like tier one that's just
sure,
develop, right?
yeah,
Single A. Then you got like a tier two. Hey, maybe you did 3 days of development. You did a single A and P run, no plan, no report for your qualification. That's what we call that, and we kind of could call that like an exploratory qual or something. And then, but it's level two, and then tier three would be, a plan, a report, 3 A and P runs, maybe 6. It's up to you. It can have a lot of gray area with all that, and then a fit for purpose like validation, and then like a validated. Sometimes you can validate a biomarker. I think there should be a somewhat of a separation there in terms of fit for its purpose, meaning, hey, I got 50% relative error on this assay, and that's fit for its purpose, right? That can be okay.
sure, yeah,
Because if
yeah,
I'm going to say
absolutely.
it's validated, that means I got to get it within that 20%. But that's just me talking, right? But I like to, I want to try and take a world of gray and try to make it a little more black and white, which is really hard to do. But John, I'll pass it back over to you. But that level tier one, I would be fine if we said, hey, we're going to perform a fit for purpose validation, and it's tier one. That could set my mindset. We just call that developed in our world, right? Versus tier two, tier three. So that's one. Stay tuned on that, John. I do think that's coming at us full bore. And then before I pass it over to you, you started to allude to it very nicely. We're talking about biomarkers, but we're going to switch over to tissues, which becomes its own kind of gray area, right? That'll be our last topic for our
Sure,
agenda today.
yep.
But tissue validations are tricky, aren't they, John? But maybe close us out on the biomarker
Well,
before
I
we
mean,
move.
the only issue I would have with when you say here's the tiers, it's like then there's going to be years of discussion
Wow. Yeah, John.
to sort of for everyone to unify on what those tiers are, because I can guarantee there'll be so many different
Yeah,
opinions about
yeah,
what tier one should look like, what tier two should look like, and there'll
yeah,
be a lot
John.
of food and phone.
Amen to that.
Sounds
You never
nice.
get it. I'm lucky. If we could get 50%, 51%, that's all you need is a majority. But amen. You're very, very right. Like I said, I desire clarity. It's
Yes,
not. It's
yes,
not. It's not
yes,
clear at all. It probably. I shouldn't say never will be, but I think there's always going to be some disagreements around biomarkers, because remember, your heart rate, your blood pressure, those are all biomarkers. How would
sure,
you
yeah,
validate
absolutely,
that,
yeah,
John?
yeah.
Like, come on.
Yeah, yeah. But I mean, but like I said earlier on, we were in this position with typical PK validation 30 years ago,
Yeah.
and we're now at the stage where everyone's pretty comfortable as to where we. I think this is a bigger topic, but
Yeah, and then you
it's still
throw in that
doable.
Cap Clear, right? You throw in Cap Clear Lab and
Yeah, sure.
diagnostics,
Yeah,
and
yeah,
biomarkers go sideways right there. If
yeah.
you're talking to someone that's trying to service a physician versus someone who wants to support a clinical trial, that you're gonna have major. I don't know if disagree. I think it's just miscommunication. I don't even think it's disagreement. I think it's miscommunication. But John, maybe let's talk tissues, right?
Yeah, I'm sort of. I'll be honest. I don't have a lot of experience in this area. I know we do a ton of tissue work, but at my previous place, we really did not. And so it's always a case of, well, obviously there's like if a drug's absorbed into tissue, ha, ha, to me the big question is how representative is an assay of a tissue? Are
Yeah.
you really getting it all out? I mean, if you're preparing um, curves and QCs by spiking into tissue homogenate, is that really representative of the tissue? Are you really
I
is
don't
that
know.
through
I think it's the
measurement?
best
Are
you can
you
do, John. You know what I mean?
or
So
or
this PK tissue,
or
right?
the final thing I would say, or is just a trend taking that approach good enough for the for tissue assessments? I don't know. To me, this is like it's like I say, I come at it as a bit of a neophyte, but I always have a big questions about what a tissue assay really represents, and But I mean, it seems we get asked to do an awful lot of them, so it yeah, to me that's the big question. I'll pass it back to you because I
Yeah.
want to throw it in
So
there because of that.
Yeah, so I think we got to always about getting the mind in the right kind of train of thought, right? So if you're doing PK versus biomarker, right, and if you're doing
Sure.
PK
Yeah.
tissue analysis, you've got some real challenges trying to mimic what the sample's going to look like in the clinic, right? Meaning,
Yep. Yep.
I got this biopsy. I can't really, it's not good practice to take that biopsy and spike my small molecule or spike my biologic into it, then homogenize it, and then go to test it. That you just lose too much during the homogenization process. It
Mhm.
just
Yep.
gets lost.
Yep.
And it's not so much that the homogenization degrades it. It's just if I put 100 nanogram in, I can't get 100 nanogram out of that because again, there's just loss. And so that's the I don't know, the innate challenge here is that no matter how good the method, you still got to do this initial step of homogenization that can cause some potential diminution of your drug, although we don't know, right? So it is common practice to homogenize the tissue and then spike the drug in afterwards, and that's as good as it gets, and that's what
Yeah.
we
And
do.
that makes perfect
And you
sense.
can go and validate that, and there's some challenges with how you validate that, but to me it's like if I'm doing PK work in tissues, I'm still going to validate the plasma. The good, one of the good practices is
Yep.
to validate
Yep.
one method, plasma, maybe CSF, just depends, but I'm going to try to validate it in some sort of more liquid like or more a matrix that's just less manipulation, doesn't require the homogenization, because then it's a little more straightforward in meeting my regulatory desire, right, John? And then when it comes to tissues, you qualify them. So you got a validated method, you qualify. The FDA loves this. It works very nicely.
Yep.
This sometimes you don't have to do it. And then when you get into like biodistribution studies, it gets a little different too, right? Like this is a deep well we're going down, John, right? Because that's not it's considered PK like, it's more PD, but man, you got to validate these assays. You don't have to validate all 25 tissues, but if I'm affecting the liver, I should validate that assay in the liver by PCR, right? And then the others I can do qualified. So just to kind of, and then small molecule has a similar kind of feel
Sure.
to it, in my opinion.
Yeah.
But if we flip over to the biomarker world, it gets a little gray because I might be able to homogenize a tissue and see my biomarker, John. Right now,
Yep.
I've got something. I
Yep.
don't. They call that an endogenous QC, right? That's the really if those that are kind of novices when it comes to biomarkers, there's a big shift in one's mind between a PK validation and again a bioanalytical validation, not clinical, but
Sure.
and
Yeah.
then the biomarker val, because when you go to do the biomarker piece, the thing you're really looking to validate is that endogenous level, a real sample, and you want to demonstrate. They call it parallelism.
Yep.
And really, that's just a fancy word for dilution linearity of that sample, but it's got you're not, see, dilution linearity means you've spiked something in by nature. When you talk about DL or dilution linearity, you've taken the sample in the matrix, you've spiked some in, and then you dilute it. Well, a parallelism is, hey, you might do that. You might do that very thing in the biomarker world, but then you want to take a live sample, a true sample that's endogenous, and then see if it can dilute. That's really the most important performance characteristic of a biomarker, in my humble opinion here. Of course, you need accuracy and precision. I don't want anybody to misunderstand what I'm saying. But if you can demonstrate or if you can get normal donors or some sort of readily available source that has your endogenous QC that can be made into an endogenous QC, you can develop a really robust validation pretty rapidly. In the absence of that, it gets tricky. Sometimes you got to get a cell line that expresses it, or maybe you got to augment a cell line to secrete something, but you got to go find an endogenous QC. I might have to procure diseased matrix in order to see it, right? There's
Yep.
all sorts of things that you have to do. So then you go from, you can take like a full on, if it's readily available in a human, I can get commercially available matrix and create an endogenous QC that I'm going to run with that. If I can't, if I have to go try to engineer one or manufacture one, I'm going to use a hybrid approach there, they call it, where I might not have 2 to 3 levels of an endogenous QC. I'm lucky if I got one. I'm going to demonstrate some dilution linearity on it, but I'm still going to use my spiked QCs as my performance characteristic, and I'll evaluate endogenous QC and parallelism only in a select few experiments, not every bioanalytical experiment. I'll pass it over to you, John. That's a lot. Hope that gave a good breakdown of how some of the things and challenges with tissues. But as you mentioned, we do a lot of it here, John, and but even as I say, hey, we know how to do this, someone will come along and ask a question and be like, gee, no one's ever asked me that, John, right? Welcome to drug development. So, but it's a fascinating thing, tissues and how to validate them.
Yeah, I mean, I'd like to say I don't have much to add because it's just not. I don't have much experience here. I do know that the big challenge for us is trying to run
what is a sample, right? What is
one, and also one tissue. When we're doing a lot of different tissues for an assay, it would be nice if we could combine them in terms of an assay that covers a lot. So you're working on plasma and trying to parallel it to tissues, but we find that a lot of the time we just can't do that. Sometimes you can combine a number of tissues within one run or one assay, but there's enough differences between tissues that the LCMS assay doesn't necessarily translate. It's more the extraction piece, and you're just not getting QCs performed the same between different matrices, etc.
That's interesting. I'm not as versed as tissues and LCMS, but it doesn't surprise me that you might need to have less ability to run standards and QCs off of a single matrix and do multiple tissues, because in the LBA world we kind of break it into like soft and hard tissues.
Yeah, and like I say, we'll try to do it, but it doesn't always work. Sometimes
it works quite frequently in our world. There's a lot of times in the LBA space you can get a soft tissue and a hard tissue kind of standard in QC, and then you just got to run the QC. And I might run 8 tissues off the soft one and 4 off the hard one, and then I just need one set of QCs for each. And I think we still, if you're in a kind of a qualified or validated, you want to put big. Sometimes it's good enough, John. You can just run buffer QCs and run your data, and you just take your baseline sample and kind of do everything off of that, right?
Yeah.
There's always, the good news is there's solutions to things. If you can't really get to the rigors you need, there are other ways to try and get your hypothesis proven with this biomarker. So
for us, I'm just going to quickly say, for us, a lot of the challenges, particularly in early or in discovery and non-GLP, will typically be using fairly generic or basic generic type extractions with protein precipitation, and it's less. You can definitely see your drug or biomarker or whatever, but it's all those other co-extractants that are different from tissue to tissue that are going to cause you problems, because that's what you're not. That's what you have to be aware of. You're not necessarily thinking about it, but that's why you have that lack of translation from one to the other. I think we're running really long on this.
That's all right, John. I'm going to finish with this. But in the LBA world, we go buy a kit. We don't worry about extractions, any of that. We just have to homogenize. So I can see why there's more universal use in the LBA world. Okay, John. Let's wrap this up.
We did have one more topic, and I think it's like emerging technologies, because obviously I'm not too affected by this, because with us it's like we're LCMS and chromatography and mass spec, unless there's there hasn't really been anything particularly disruptive on that, because you're using the chromatography is the same, the extractions really much, we're using lots of different approaches, but still relatively the same, and then the mass specs are all we're using as glorified detectors, but they're still relatively the same. But with you, I mean, we got obviously the new Lisa that we're installing in Lyon. That first of all, we're bringing it in. It's like non-GLP, but it's a different approach. So, and I don't know enough about the approach to really say, but what's the approach with a new technology? How do you get that into the more validatable frame of things, or how do you decide if it's always going to be non-GLP versus we can get it into validation? I was just interested in your thoughts on that.
Yeah, so the new Lisa is an immuno PCR system, John, right? And really cool stuff. It can, in one like 20 microliter or 10 microliter sample, you can get almost 250 biomarkers out of it, or 120. They got these massive plexes. But to answer your question, and it's a good one, how because that platform has only been recently maybe validated. I don't even know if it's truly IQ, OQ, PQ'd, and we're trying to do that. I think there's 1 or 2 other CROs trying to do it, but nobody's, to the best of my knowledge, actually run a true validation on that instrument or tested thousands of samples on it in a regulated setting. And you kind of ask, well, what goes into that? And John, there's 2 major things in my opinion. That is it quantifiable? If the system itself is a semi-quantitated system, it's one that I'm a little, kind of, man, that's going to be a challenge, right? Meaning, Olink, for instance, is something that people say they do regulated work on, and then they'll tell you, well, it's semi-quantitative, and that, man, that's hard to explain to a regulator. Some regulators are okay with that. It's like using the word PK-like, John. Those are words we just don't like saying.
Sure.
Semi-quantitative
Yep.
and PK-like are really, if you're PK-like, it's a PD assay. Let's call it what it is. And if you're doing something semi-quantitative, then that really should be put into a qualified area, not a validated one, John. In my word, but again, that's where some of my European colleagues would think I just said heresy there, John. Right? Because that still can be validated, right? So it's just shades of gray, as the topic should have said, the gray zones. And so to round out your question one, if it's a quantifiable instrument, that puts it in a higher, to me, it's like, okay, that makes sense, right? And that's pretty straightforward. The other, John, is the CFR 21 compliance and the way you regress the data in the computer. And I speak about this because we were the first lab to validate the Quanterix Samoa system, and we had to educate them on how to do that, John. We leveraged our 40 years of bioanalysis experiments because they were just trying to build a box that was ultra sensitive, right?
Sure.
They didn't understand a 4-point parameter fit, a 5-point parameter fit, the regression through zero, all these things that go into really critical things. For bioanalysis. So, it's a great question. I hope I gave you a clear answer, right? It's what is its computer kind of, is it, and when I say CFR 21, that's all, how secure is that chain of custody of the data?
Sure.
How secure is it? A lot of people don't think about that. They're just like, oh yeah, you can change a number in this. Well, that is not something we would want to put in our laboratory, right? It's got to have that sort of locked. After the data comes off the box of the machine, it's got to be in a state that cannot be manipulated, right?
Sure.
Absolutely. So that requires some IT in there, and then that quantitative piece. But those are the boxes, people that the NULISA, it seems like they're ready at the NULISA, although I've talked to a few there. They're still trying to figure out that PQ component of the IQ, the installation, operation, and production of it. The PQ of it becomes performance. Sorry, I said production. Performance of it. Thank you. But that becomes, I think that's iterative at this stage. That's evolving at this stage for the NULISA system.
Yep. And there's your challenge with new technologies. It's evolving, it's not clear. So you're having to stepwise figure out your way through it.
But John, if you buy a mass spec or an MSD instrument, they sell you an IQ, OQ, PQ package.
Yeah,
right. Yeah. NULISA doesn't have that just yet, right?
Yep.
That's and then now, Quanterix may have one, but we kind of helped them write it at the time. And John, it took a long time, and it was really, anyhow, making me shake. And there's a few other systems that I've done that on here. It was something called a Genelite system, and so we've got some experience here at KCAS Bio. But we're happy to leverage that experience and really are looking forward to this NULISA system. It's wonderful, John. It has incredible application to it, and they have made their 120 plex quantifiable. It's just a big investment to do that.
Sure, absolutely. Yeah. Okay. Any final thoughts before we wrap up? Because I thought this would be a light topic, and we've gone way long.
No, as soon as you said it was gray zones, I'm like, I live in the gray. No, I want to be black and white. But no, John, that's it. How about any big plans coming up in the next couple of weeks or this week?
Yeah, so next couple of weeks, as of 7:00 tomorrow night, I fly back to the UK. Or my wife and I fly back to the UK, but as usual, we fly separately. She's gone mid-afternoon. She flies Delta. I fly American, so I'll fly out at 7. And then we meet at Terminal 3 in Heathrow. We then get the train out to Norwich, which is where my wife grew up. Spend a few days in Norwich. Actually, the first 3 days were in the hotel where we spent the first night of our marriage. And nice. Then after that, we go and visit our sister because her husband, it's his birthday on the Sunday, and he hits a milestone that none of the rest of us have hit. Hopefully we all will. He turns 70, and so he's Sunday. Their daughter turns 18 on the Monday, and then my wife's sister I'm not sure what age she turns on the Wednesday, and then we fly back on Thursday and Friday of next week. But I can't wait to do it, because like Norwich is a really nice city.
Yeah.
Its city motto is Norwich, a fine city. It's more than fine. It's a lovely place, and
I believe they're the Canaries in the soccer, right? Are they the
yeah,
Canaries?
exactly. Yeah, yeah, and it's like, because our travel has been, both of us do a lot of traveling, but we do an awful lot separate. So Kath has been back to Norwich multiple times, but I haven't. So I just, when we knew it was like Philip's birthday, we thought, let's make a trip of it and actually go and have just a really nice few days. And so yeah, can't wait.
Yeah, that sounds super exciting. I love the fact you're going to go spend a night in your where you got married. That's really cool. I got married in Columbus, Nebraska, and periodically we drive by it, and we've never stayed there again. Although it's a big like a nice bed and breakfast, so it's got me.
So I do have a story about the honeymoon suite in this hotel. It's called the Maid's Head, and it's just it's not far from the cathedral in Norwich. But the core of the hotel is really old. I think it's Tudor, so like 15th, 16th century, maybe 16th century. I'm not quite sure with the origin. But the honeymoon suite was, well, at least when we went there, which is a long time ago, is fascinating, because the main bed was this 4-poster bed, which was, I mean, I think probably 3 foot off the floor. So you needed steps to get into the bed if you're small.
Yeah.
Like some people. But there was actually a corner of the room. If you stood in the corner of the room, it was higher than the level of the mattress of the bed. So it tells you what the shape of the room. It was fantastic, you know. So yeah, we're not staying in the honeymoon suite, but it'd just be fascinating to go back there.
It's funny, the honeymoon suite in Columbus was maybe I think the building was like 100 years old, not as old, but it did have you did have to step on a bed to get. You had to step on some stairs to get in it. It was really high.
It's like that common theme. So that's basically it. We'll finish packing today, and we'll be set to basic counting down to both of us flying out. You know, we've both got our shopping lists ready for what we do, because it's like lovely, you know, great secondhand bookshops and just new bookshops and record shops, and I just really, it's just a great city. So yeah, I'm really looking forward to it. So what about you?
I got something similar. It seems like there's a lot of birthdays in the summer. So my mom and myself have birthdays first week of August. So not leaving right away, but August 1 through 7, I'm in Boston. Got a couple nights in the harbor in Boston. We're going to stay downtown. My son and his girlfriend are coming with us, which is the first. So that's big. That's big there. But the 4 of us will spend a couple nights downtown. Gonna maybe cruise around and do some. Might go on a whale watch. We do a duck boat tour through Boston, and you know, my son doesn't really remember it. But you know, we'll have a fun time doing some standard Boston tour stuff. Maybe go Paul Revere's route, or go eat in the North End and do some shopping and all sorts of fun things. But then the big thing is we'll go down to Cape Cod for 4 nights. We rented a big old house with my sister and her family. There's about, I think, 10 of us staying there. My brother's family will come down, and my mom turns 80, and we're going to celebrate her 80th on her 80th birthday, which is August 6th. And she's a big Elvis fan, so my sister's been buying all this Elvis stuff, and we're going to – place has got a karaoke machine. It's a big house we rented, right? It's not right on the water. It's about a 2, maybe 5-minute walk from the water. Got a nice pool in the backyard. So we're pretty excited to go spend 4 or 5 days down in Cape Cod, and – haven't been down there in a long time. We might try to go see Nantucket, or actually I'd probably go see Martha's Vineyard, not Nantucket, and you know, just tool around there for a little bit, and more importantly, get to see my mom dance to Elvis. She's obsessed with Elvis these days, which is hilarious. But yeah, no, both of us got some fun stuff going on, John. But as you said, this went a little long. Let's wrap up. We'll see you in the funny pages.
Yep. Thanks very much. That was a lot of fun. Thank you.